Thanks, Frank. Take it away.
Thanks very much for the extra privilege of being able to address the SB Grid community 2 weeks in a row. Last week, we talked about some general details of signal processing. Today, we'll see how things work under the hood in the NMRPipe system. We'll see the details of starting with raw data from the Spectral, how to convert it to NMRPipe format, how to inspect it, choose phase correction parameters, and build processing schemes. Then we'll see how to display spectra and to generate peak tables from the spectral data. The first part of the demonstration will show all the manual methods for carrying these steps out, and then at the end, we'll have a glance at methods that automate all of these steps. All of the aspects of spectral processing in NMRPipe are conducted either directly or behind the scenes by the use of scripts. And the scripts are either C-shell scripts or TCL scripts. In order to process spectral data with NMRPipe, we start by creating a script that converts spectral data from the spectrometer format to the NMRPipe format. And we have a simple graphic interface to help us with these steps. It's the same graphical interface for all types of data, but we invoke it a little differently depending on whether the data is from a Bruker instrument or from a Varian and Agilent instrument. And when we invoke this conversion tool, we get a pair of windows like this. At the top is the graphical interface, and at the bottom is a little text editing area where the script itself will appear. So as a first step, I'm going to use the file browser to select the spectrum that I'd like to convert and process. In this case, we're going to work with a 3D HNCO experiment. Once I've selected the data to convert, I'll use this button in the interface to read the parameter files from the spectrometer format. And at this point, the interface will do its best possible job to interpret the various parameters in the spectrometer parameter files in order to identify the sizes of each dimension in the spectral data and the chemical shift calibration information. Now, depending on how the spectrum was measured, the parameter files may not actually include unambiguous information to define these parameters for each of the dimensions. So we often have to give them a little extra attention. And although maybe you would say I'm a little biased about this, data from a Bruker spectrometer needs a lot more special attention than data from an Agilent spectrometer currently does. But we'll see more about that. So in practice, what we do most keenly is establish the chemical shift calibration information. And I'm going to talk about this in a little detail because it's particularly important. The chemical shift calibration is established on the assumption that the center of the directly detected dimension is at the chemical shift of water. And the interface will read the acquisition temperature from the spectrometer parameter files and use that to calculate what the chemical shift of water would be at that temperature. Given that result, this chemical shift calibration information from the directly detected dimension can be propagated to the other dimensions of the spectrometer. However, in many cases, the values recorded in the spectrometer parameter files are not actually accurate representations of what really happened during acquisition. Most commonly, especially on Bruker spectrometers, the temperature value that's recorded in the parameter files may bear no actual resemblance to the actual temperature that was used during measurement of the spectral data. What can we do about this? Well, let's say we've seen that the spectrometer parameter file has said that the temperature is 300. But in fact, let's say instead that we know that the real temperature is 278. So I'm going to type in the correct temperature here by hand, 278, and I'll return to the graphical interface. Right now, the center position of the x-axis of the data, the directly detected dimension, is set to a particular chemical shift value, and instead I'm going to set it simply to the keyword H2O, which means interpret the center position as if it's at the chemical shift of water. And now when I read the parameters a second time, it will recalculate the chemical shift of water based on the temperature I entered manually and propagate that chemical shift to the remaining dimensions of the experiment. The other information that's critical but not always recorded unambiguously in the spectrometer parameter files is how the channels of the spectrometer are mapped to the dimensions of the experiment. So we often have to select that information manually, which we do by going to the menus for the observed frequency in each particular dimension and selecting which channel of the spectrometer is associated with that dimension. So in this case, for an HNCO, it just so happens that the second dimension is the carbon dimension, so I'll select carbon from this menu. And the third dimension is the nitrogen dimension, so I'll select that. And then finally, if we wish, we can give specific labels that relate to the chemical information that's associated with a particular dimension. So in this case, for an HNCO spectrum, I'll rename the carbon dimension CO for carbonyl. And you may have noticed that as we make these adjustments in the graphical interface, the corresponding shell script at the bottom gets updated automatically. This shell script is what will actually be executed to convert the spectral data from the NMRPipe format, from the spectrometer format to the NMRPipe format. So I'll save this script. And now execute this script to convert the spectral data. Now I've done this from within the graphical interface, but if we return to the Unix command line, we can actually type the contents of this conversion script on the screen, and we can execute it directly from the Unix command line by typing in the name of the script. So now let's inspect this raw data, which we do with the program called NMRDraw. This is what the first 2D plane of the 3D time domain data looks like. In this version of the display, the areas where the time domain data is intensely positive are drawn in blue. The areas where it's intensely negative are drawn in gray. And the areas where it's close to zero are in black. The command panel of NMRDraw has a collection of menus. One of them, the mouse menu, is a list of all the things that you can do interactively with the mouse. So at first, I'll select a command called 1D horizontal. And that will let me view one-dimensional slices at different positions in this 2D plane. What I'm doing right now is pressing and holding the left mouse button and dragging it through the spectrum so that I can see different 1D slices. And if you look closely, you may notice that as I drag through the slices, the slice number is displayed in the command panel. And I can also use these caret buttons in the command panel to advance the slice number one at a time, or to type in a particular slice number that I'd like to see, like for example, slice number 1. Just type in the number 1 here. So here's slice number 1. And when a particular graphical mode is turned on in the NMRDraw program, the topmost border of the program tells us what the different mouse buttons do. And depending on whether the mouse pointer is inside this spectral graphics area or outside in the borders where the axis is drawn, the buttons of the mouse can potentially do different functions. So for example, according to the help text in the window, the middle mouse button can be used to change the vertical scale of the current data, and I'll do that now. And the leftmost mouse button can be used, the rightmost mouse button can be used to change the vertical offset of the data. So here's the first 1D vector from the first plane in the time domain data. We can use this to choose phase correction parameters, but of course we have to turn it into a spectrum. And a convenient way to do that is in the process menu through a command called auto process 1D. And that will apply a predefined collection of processing functions that include a window function, zero fill, and a Fourier transform. And we get a result like this. We'd like to choose phase correction for this. We can do that in the command panel as well. There are buttons in the command panel that will turn interactive phasing on. So I'll click those with the mouse. And when interactive phasing is on, we can use these slider bars in the graphical interface to adjust the zero order or first order phase. And I can do that either by dragging the slider bar or by clicking the mouse on one side of the slider bar or another, and that allows me to increment or decrement the phase by one step at a time. Each phase correction has two slider bars, one for coarse adjustment of the phase and one for fine adjustment of the phase. And the phase value itself is reported on the screen. And we can also type in a particular number and apply that phase correction correctly. We'll see more about how to phase or rephase already processed data. But at this point, we have the first 1D vector from the data. We have a reasonable phase correction. So now we can build different schemes to process part or all of the spectral data. And as we mentioned before, this processing is done by creating shell scripts. We can create them with a basic text editor, but the NMRDraw program also has a text editing facility inside of it that will help us create appropriate scripts to process the spectral data. And I can find that text editor under the File menu in the option called Macro Edit. The Macro Editor is a point-and-click text editing screen that has a collection of menus that will help us build most commonly used types of processing schemes. So I'm going to erase the scheme that I have here and go to the menus and choose an option to create a scheme to process 2D data. In this case, I'll choose the scheme to process the first XY plane of a 3D spectral data set. And when I choose that, the text editing area will be populated by the appropriate script. This is a very typical kind of NMRPipe processing script. where the NMRPipe program is used over and over again in a long pipeline to apply a collection of different processing functions. At the beginning of the pipeline, we read data in from the time domain input, and at the end of the pipeline, we write the processed result out to a frequency domain output. And in between, we apply the different processing functions that we'd like to use. In this case, for 2D processing, this particular scheme uses a solvent filter, a window function with scaling of the first point of the time domain data, a zero fill, a Fourier transform, a phase correction, and extraction of a particular chemical shift range of that dimension of the data. Then we have a transposition function that allows us to start processing the second dimension of the data. And in that dimension, we apply a window function, a zero fill, a Fourier transform, and another phase correction. So the first time you see scripts like these, they can be quite overwhelming, but the general learning curve of NMRPipe is becoming familiar with these most commonly used functions and the command line arguments that we use to set their parameters. The ones that we change most commonly are simply the phase correction values. And so I'll point and click and insert a particular phase correction value in the script, and then save the script, and I can execute it from inside this interface to produce some spectral data. And now I'll use an option in the file menu that will give me a file browser so that I can find the process result. So there's our spectral data processed by that scheme. And the same way that we looked at vectors from the time domain data, we can also look at vectors from the frequency domain data, either along the horizontal axis or along the vertical axis. And if we need to choose phase correction parameters for the indirect dimension, we can take any one vector and tell NMRDraw to keep it on the screen, and take as many other vectors as we like from elsewhere in the spectrum. And what this enables us to do is to see several vectors at once when we choose phase correction values. So this is a nice convenience. I can also use the mouse to adjust the contour levels for this display. I'm scrolling with the mouse wheel now. And as I scroll down, and we can start to see what's happening in the baseline of the spectrum, I see areas that are predominantly red or predominantly blue. And if we look at 1D profiles, look closely at these 1D profiles, what we'll see is that particular rows of the data have a baseline offset. So this blue line is where 0 is, and the spectral data in this particular row is substantially below that 0 line. So what that means is that there are systematic baseline distortions still left in this spectral data, left over as a result of the solvent signal that was in the original time domain. So as an example, let's go back to our processing scheme and add some steps to perform baseline correction, which we will do after all the spectral processing is done. And so I'm going to add an additional NMRPipe function here to transpose the data back to the directly detected dimension. And then I'll add an automated baseline correction in that dimension. And I'll save the result. process the data. Okay. So here's the spectrum before, and here's the spectrum after. So now there's no systematic positive or negative offsets in different parts of the spectral data. Very easy. So that's a scheme that we can use to process the first 2D plane of the spectral data. Let's see what it's like to build a scheme to process the entire 3D spectrum. And I'll do that from the Process 3D menu. And this time I get a processing scheme that has two parts. The first part is very much like the 2D processing scheme that we built before. And the second part is a scheme that processes the remaining third dimension of the spectral data. So I'll use this scheme, and it's counting through all the planes as it processes the first two dimensions. And now it's processing the vectors in the remaining dimension. Let's read in the processed result now. So here's one plane in the interior, and I can scroll the mouse wheel to jump through the different planes of the 3D data. And just as we did before, we can look at vectors from the horizontal dimension of the data or the vertical dimension of the data. Or we can look at vectors from down along the third dimension, the z-axis. So in this case, I can point to a particular position and see the vector down along the plane. And then finally, if I'd like to perform peak detection on this spectrum, very simple. I use the peak detection pop-up, set the number of dimensions to 3 so that I can do peak detection on the full 3D spectrum rather than this particular 2D plane, and press the peak detect button. And in a few seconds, I have a complete peak table for the 3D data. And as I scroll through the spectrum, I can see the peak labels. And if you look closely, what you'll see is that the peak labels have a few different formats. Some cases, the peak label is just a number, but in some cases, the peak label has a greater than sign or a less than sign at the beginning. This means that the peak maximum for that particular peak is at a future plane in the data or a previous plane in the data. And then finally, some of the peaks are labeled with an asterisk instead of a number. That means the peak detection program has decided that that peak is most likely due to either random noise or that peak is a truncation artifact of a nearby larger peak. For example, let's see. This small negative peak here is actually a truncation artifact of a larger peak in the next plane. Very simple. And the peak detection program records all manner of parameter estimates for the peaks, which we can display on the screen, like the chemical shifts in a particular dimension. or the height of the peak. All that's saved in a simple text table. So those are the basic steps for going from raw spectral data to a finished spectrum and a peak table. And the scripts that we used are all saved on the disk as text files. So they form a complete record of everything that we did to generate a particular spectrum. And not only are they a complete record of what we did, but they can actually be used directly to recreate the spectral result if we ever need to. So this is an example of the 3D processing scheme that we used. But we can also take a 3D scheme like this one and add a recipe for displaying the result. And this recipe builds a 2D projection, detects the peaks in the 2D projection, and displays strip plots at all of those positions. So this is also another nice way to get a complete overview of a processed 3D spectrum. And here's what that result looks like. So in this display, I see pairs of strips at all the positions that we were able to find the peak in the 2D projection. So this is a nice way to get an overview of the entire 3D spectral result and see what the line shapes look like in all three of the dimensions. And we can use this strip viewer to view more than one spectrum at a time. Before we started this demo, I executed a couple of other processing schemes that process the data with linear prediction, which we discussed last week, and also with maximum entropy. And now I'll use a script to invoke that strip viewer to display these results. And in the strip viewer, we can grab particular strips and reorder them. Now, as I mentioned, these steps can also be automated. And what I'd like to do, almost as a little commercial, if you'll forgive me, is show you how these steps are automated on Agilent format data. In this case, all of the steps of creating the different conversion and processing scripts are handled automatically. So there's no need to edit or adjust a script. The conversion script is built automatically. Scripts that process the first XY plane and the first XZ plane are built and executed automatically. And then the full 3D data is processed with the same schemes that we saw: the direct dimension, the indirect dimension, baseline correction. and then finally the remaining third dimension. And the steps that we saw before to generate strip plots of the spectral result are executed automatically. So that with a single command, we go directly from raw spectral data to a fully processed result. So that's my quick overview of basic spectral processing in NMRPipe. I hope it was interesting for you. There are many other examples that use processing methods like linear prediction, iterative soft thresholding, and you can find those in our documentation.
Great. So I'll sort of kick off with a question related to that. If users want to find documentation, what would you recommend as a sort of go-to source for documentation on NMRPipe?
Actually, NMRPipe is widely used enough now that you can simply use Google to find things. Like, for example, if I want to learn how to do linear prediction in NMRPipe, I can't start a web browser because of the particular graphics environment that I have going on. But if you just say NMRPipe LP, you'll find the NMRPipe documentation for linear prediction. And in particular, for people who use Agilent and variant spectrometers, we also have the Spin Sites forum, which has additional materials that are specific to data from Agilent and variant spectrometers. So if you're not a participant in that Spin Sites forum, it's a nice place to join to find information and examples like that.
And can you process data that is non-uniformly sampled in NMRPipe? Is there support for those algorithms, techniques?
There is, but right now it's only for variant and Agilent data.
Okay.
However, that said, all of the groups that produce software to process non-uniformly sampled data, like, for example, Gerhard Wagner's group and Sven Hebert's, have versions of non-uniform sampling that work with NMRPipe format data. So, in fact, most of the solutions for processing non-uniformly sampled data actually require NMRPipe format data in order to work. So there are many examples and pieces of software available to do that.
It was really great. If anybody has questions, feel free to type them in the chat. We can answer them. I guess with that, we can call it. I thank you very much, Frank. I really appreciate it. I think it's really informative, and I know my Spectra always look best in NMRDraw for some reason. I don't know. It's just for some reason the display, the contour maps, they always look great. Then I switch over to something else, and I think like, oh, okay, it looks better in NMRDraw.
Thank you.
Great. Thank you very much.
Thanks, everyone. Bye.
Bye.